Presidential Professor at the University of Oklahoma Health Sciences Center and Associate Director for Translational Research at Stephenson Cancer Center at the Oklahoma University Health Sciences Center. He earned his master's from University of Burdwan in '94 and a PhD from University of Pune in 2000, both in chemistry. From 2001 and 2003, he served as a postdoctoral fellow at Texas Christian University. He later completed his fellowships at Harvard Medical School and then at Mayo Clinic, Rochester, Minnesota. In 2005, Mukherjee became an assistant professor at the Mayo Clinic in Rochester, Minnesota, and was promoted to associate professor in 2009. In 2013, he left the Mayo Clinic and joined the OU Health Sciences Center as a full professor in the Department of Pathology at the OU College of Medicine. Professor Mukherjee is a fellow of Royal Society of Chemistry, the American Institute for Medical and Biological Engineering, and the National Academy of Inventors. In 2018, he received the Outstanding Achievement Award from the Society of Asian American Scientists in Cancer Research. The floor is yours, sir.
Okay, thank you so much, Mehmet, and thank you, Ali, and thank you all for joining for today's talk. So as you can tell from the talk, it is cell-nanomaterial interactions, implications in biology and medicine. So the theme of the talk is, again, as you can see, it is little underrepresented area of research. So here my purpose is to tell you three short stories and to demonstrate how really nano can help us to understand the biology of any disease better. And those who have not been to Oklahoma, so this is our cancer center, Stephenson Cancer Center. We got NCI designation in 2018. And we are going for the renewal this year, so we are kind of busy towards that. And as Mehmet said, I serve as the Associate Director for Translational Research in our cancer center. So now coming back to the topic, so let me take you back in the early part of 2008 and 2009. So where we had been trying to understand three fundamental issues related to nanomedicine or nanomaterials and how it really influences the cell behavior. So why is it not moving? So Mehmet, why it is not moving?
I'm not sure. If you maybe get out of this mode and try again, or usually it should move. John, are you there?
I got it, I think, yeah.
Okay.
Okay, great.
Sorry about that. So within the theme, so I will talk about surface engineered nanomaterials. We will talk about how receptor targeted drug delivery system. And we also talk about something called cell therapeutic nanoparticles. And we'll try to understand how each of those nanoparticles interact with the cells and what information we can get from those interactions about the biology of the disease process. And as you can tell, our cells of interest are obviously cancer cells, endothelial cells, and fibroblasts. And as you know, those are the three major cell types in the tumor microenvironment. And as we progress along the seminar, you will realize or you'll come to know how really nanoparticles can help to disrupt the multicellular crosstalk that goes on in the tumor microenvironment and how we can understand the molecular intricacies that play out and identify the key perpetrator. So with that, let me start with the surface engineered nanoparticles. Again, all of the data that I'm going to show here, we already have published. So I'm not going to really bombard the screen with too many data, but I'll just give you enough information just to support the concept. And rest of the materials is already published. We can get it out from the published paper, or we can get onto that during our discussion session. So the first short story is surface engineered nanoparticles. So as I said, early 2008, 2009, we were trying to understand, so if we create nanoparticles of different charges, say positive, negative, deuterionic, and neutral, and if you throw them to the cells, how really the cells are going to interact. So that's what we started. We didn't know what to expect, but we thought we will follow the process as we go along. And so what we did, and again, this is in collaborations with Vince Rotello, who is a professor at UMass Amherst, with Prakash, who is now the chair of biomedical engineering department in our previous institution, Mayo Clinic, and Rosal Arvizo, who was a postdoc at the time, who graduated from Vince Rotello's lab. Again, so we wanted to know how the cells will respond if we treat cells with this type of nanoparticles. So this is neutralized charges around -1 millivolt. This is highly positively charged nanoparticles, and it is plus 24 millivolt. This is deuterionic charge is close to neutral, and this is anionic, which has a high negative charge. So now you have these nanoparticles, and you are throwing it to the cells, and how the cells will respond. So that is the questions that we wanted to answer. But as you know, cells really interact with the extracellular environment through their plasma membrane. So we thought, let's probe at the plasma membrane level what happens if we throw these materials into the cells. And again, as you can see, I published this paper in Nano Letters in 2010. It's almost a decade ago. So when we looked at now the plasma membrane potential, and again, this is in collaboration with Prakash at Mayo Clinic, what we did find out is that positively charged nanoparticles depolarize the plasma membrane. And again, from the charge of -70 millivolt, it comes down to -30 millivolt. But it happens only with positively charged nanoparticles. And again, if we treat the cells with standard depolarizing agent, which is potassium chloride, we do see the depolarization to the same extent. But however, when we treat with the other nanoparticles which are not positively charged, there is actually nothing happens to the membrane potential of the plasma membrane. So these are CP70 cells and 2780 cells. Those are the cancer cells. And this is human bronchial epithelial cells and human airway smooth muscle cells. Those are the normal cells. So what we see from here is that irrespective of whether those are cancer cells or normal cells, the plasma membrane of those cells were depolarized when we treat them with positively charged nanoparticles. But other nanoparticles do not have those depolarizing capacity. Now, what are the consequences of this membrane depolarization? Basically, you are removing kind of electrical barrier. So the expectation is the lot of materials which are outside in the extracellular fluid, they are going to come into the cells. So we thought we'll look for the calcium first, because as we know, calcium is also regarded as second messenger, and we'll see what would be the biological consequences of the membrane depolarization. So what we found is that when we added the nanoparticles here, the membrane depolarization starts, and what we see on the other hand, that calcium level inside the cells increases. So as the membrane goes down from more negative charge to less negative charge, you get calcium influx inside the cells. And the consequences of that enhanced calcium entry into the cells is that there is apoptosis in the cells. And those apoptosis happens only in the normal cells. But the CP70, as you can see, the black bar, they remain viable, they remain alive, but only the normal cells are dying, these bronchial epithelial cells. And the same thing happens with, again, airway smooth muscle cells as well. But again, if we look at CP20 and A2780 cells, these first two bars, we don't see any changes either in proliferation. So these are the proliferation or in the apoptosis. So basically, the consequences of the membrane depolarization is increase of calcium influx into the cells, and the result of which is cancer cells are not dying, but the normal cells are dying. Now, why the cancer cells are not dying, but the normal cells are dying, when the level of calcium entry into the normal cells as well as the cancer cells are same? So we thought, let's try to figure that out. And so also we wanted to know, okay, what are the source of that calcium coming from? Is it really coming from the extracellular medium, or is it coming from the stores within the cells? So what we did, we used those blockers. This is nifedipine, which is L-type channel blocker. And this is zexospondin C, which is IP3 receptor blocker. And also we use ryanodine receptor blocker. So what we are seeing from this inhibitor experiment is that that part of the calcium is coming through L-type channel, because when you use nifedipine, which is L-type channel blocker, we see there is a decrease in calcium amplitude within the cells. Similarly, when you are using zexospondin C, which is IP3 receptor blocker present on the endoplasmic reticulum, we also see there is a decrease in total calcium level inside the cells. So the conclusion from this membrane depolarization and increase in intracellular calcium influx is that those calciums are coming from outside as well as from the endoplasmic reticulum. But even if that is the case, but why the cancer cells are not dying but the normal cells are dying? So it seems like cancer cells has something to overcome that calcium influx into the cells. So now if we look at that entry of calcium mechanism and the different stores inside the cells, there are mainly two places where calcium can be stored. One is endoplasmic reticulum and one is mitochondria. So now imagine, so when you are treating these cells with positively charged nanoparticles, this membrane probably are getting, again, it is getting depolarized, and calcium is entering into the cells. That's through L-type channel, as we have shown through L-type channel blocker, and some of the calcium stored in the endoplasmic reticulum, they're also coming out. And the resultant is increase in total calcium level from 100 nanomolar to around 500, 600 nanomolar. So 5 to 6-fold increase in calcium. So under that condition, the normal cells are dying, but the cancer cells are not dying. So why? So we thought maybe cancer cells have some mechanisms to withstand or bypass these calcium entry into the cells. And so we are trying to figure out what that mechanism could be. And during that time, but before that, if you look at the history of mitochondrial calcium research, so since in 1961, it was known that calcium gets into the mitochondria. But until 2010, people figured out in 2010 what is the machinery that regulates calcium entry into the mitochondria. And 3 papers came out back to back in the same 2010 when we were carrying out those investigations, is that the MICU one and then MCU. So these are the basically machineries or proteins which are present in the mitochondrial inner membrane that regulates calcium entry into the mitochondria. MICU1 in particular acts as a negative regulator. So basically it prevents calcium entry into the mitochondria, whereas MCU is calcium uniporter. It promotes calcium entry into the mitochondria. So we, based on this information, we thought maybe MICU1, which prevents calcium entry into the mitochondria, is regulating that cell death in normal cells and cancer cells. So what we were thinking is, if there is overload of calcium into the mitochondria, cells undergo apoptosis. And what we know from our data is that normal cells undergo apoptosis, but cancer cells do not. So maybe then in the cancer cells, it has some kind of barrier that prevents calcium entry into the mitochondria. And so we were trying to figure out what that barrier is. And we've hypothesized, based on these publications, probably the barrier is MICU1. So this is a protein which is present in mitochondrial inner membrane and which prevents calcium entry into the mitochondria. But whether MICU1 is playing a role or not, so we wanted to test. So first we looked at the expression of MICU1 in normal cells versus different cancer cells. Again, since it was just discovered, so there was no antibodies against MICU1, so we probed it through real-time PCR analysis at the messenger level. And what we do see here is that compared to normal cells, MICU1 expression is high in all of the cancer cells that you have tested at that time. And interestingly enough, when you knock down MICU1 using siRNA and then you treat with positively charged nanoparticles, you can see like the cancer cells are now dying. Whereas when you have RU360, which is a chemical inhibitor which blocks calcium entry into the mitochondria, nothing happens with the cell. So basically, then if you can remove the barrier somehow and allow calciums to get into the mitochondria, then you can make cancer cells die. But now the interesting part about these normal cells and cancer cells are that in the cancer cells, the MICU1 expression is high, means the barrier to calcium entry into the mitochondria is high, whereas in the normal cells, the barrier is low. So those calciums that got into the mitochondria that got into the cells after treating with positively charged nanoparticles, for the normal cells, they can easily get into the mitochondria, but for the cancer cells, because of the barrier, it cannot get into the mitochondria, and that's why cancer cells survive, they do not die. But if you remove that barrier by means of silencing MICU1, which is the barrier, then you can see that you can really now induce cell death to those cancer cells, which were then resistant. So again, we published these two papers. This in JBC paper came out in 2013, and now figuring out what exactly MICU1 does to the cancer cells. But that gave us a starting point. So again, as you have seen in the cancer cells at the messenger level, the MICU1 is high. And now the next question is truly how really is MICU1 making cancer cells resistant to apoptosis? So then Prof joined in the lab. He's now a senior scientist in Google. So he thought, okay, so he will take up the challenge and to figure out really what MICU1 does to the cancer cell. So now by the time we got the antibodies for MICU1, and as you can see between ovarian surface epithelial cells, the normal cells, or fallopian tube endothelial cells, which is also normal cells, MICU1 expression is high in all the cancer cells. Then we wanted to go back to the data sets, patient data set, to see whether high MICU1 expression is correlated with poor prognosis in ovarian cancer. And as you can see, whether MICU1 low versus MICU1 high, the green versus red, when MICU1 low, patient behaves better in terms of survival, they survive longer, versus when MICU1 is high, their survival is poor, so they do not survive much longer. And to further bolster it, because again, these are all at that gene level, RNA level, and always you can see there may be some discrepancy between RNA expression versus protein expression. So we created our own tissue microarray. So where we have grafted 125 patient tissues from the high-grade serous ovarian cancer in a slide, and then we looked at the expression of MICU1. So as you can see, MICU1 expression goes from weak to moderate, and what we have found from the analysis is that almost in the 80% of those drug-resistant high-grade serous ovarian cancer patients, MICU1 expression is very, very high. So whatever probably we have found that MICU1 correlates with better survival of the cancer cells probably is true, and that's why when MICU1 is high in the cancer cells in the patient tissues, and we see the patient behave poorly. But how this overexpression of MICU1 really makes cancer cells to survive? So what we figured out is that, again, as I told before, I'm not dumping too much of data here, just I'm giving you the crux of the paper, again, that you published in Nature 2017. So the key here is, so what we have found is MICU1 actually is a glycolytic switch in ovarian cancer. And how it does by regulating calcium entry into the mitochondria. As we know, glycolysis, or the Warburg effect, is the extramitochondrial utilization of the glucose. And extramitochondrial utilization of the glucose is dependent on two enzymes. One is PDK, which is pyruvate dehydrogenase kinase, and the other one is PDP, which is PDP, pyruvate dehydrogenase phosphatase. What it does, glucose gets converted to pyruvate, and then pyruvate to pyruvate dehydrogenase. This pyruvate dehydrogenase is the substrate for mitochondrial oxidative phosphorylation. But when it gets phosphorylated by PDK, pyruvate dehydrogenase kinase, then it is no longer a substrate for mitochondrial oxidative phosphorylation. But this phosphorylation on pyruvate dehydrogenase is stripped off by PDP, which is pyruvate dehydrogenase phosphatase. Now, the enzymatic function of PDP is dependent on calcium. How much calcium is there into the mitochondria, that dictates whether PDP will be functional or not. So now think about the normal cell and cancer cell context. In the normal cell, because the MICU1, the barrier is low, so there is enough calcium into the mitochondria, and then PDP is functional. then it removes that phosphate backbone from the pyruvate dehydrogenase and makes it again the substrate for oxidative phosphorylation. And that's why normal cell is dependent mostly on oxidative phosphorylation. Whereas in the cancer cells, the MECO1 barrier is high because the expression is high. So there is minimum entry of calcium into the mitochondria. So because of which the PDP function is low, it cannot take out that phosphate backbone from PDH, and that's why it cannot be used anymore as the substrate for oxidative phosphorylation. And that's why glucose is utilized extramitochondrially. And that's what we figured out in this paper in Nature Com that we published in 2017. But again, we did not stop there. And then as we have seen in that tissue microarray and also in the cell line data, MECO1 expression is very, very high. And then the next obvious question was why MECO1 expression is high in cancer and low in normal cells. And we also figured that out. So there is a particular microRNA, which is microRNA 195, that actually controls MECO1 expression in ovarian cancer, and that regulates tumor growth. And so the story here is we started to understand really how those positively charged nanoparticles or negatively charged or zwitterionic or neutral will interact with the cells, or on the other hand, how the cells will respond when you treat the cells with those type of nanoparticles. And that understanding, or trying to understand that mechanisms, led us to discover a molecule MECO1, which is now a glycolytic switch in ovarian cancer. So that is, I think, the beauty of nano. So if you try to challenge this nano with the biology and try to understand how nanoparticles interact with the cells, we really understand something new, which otherwise we could not have understood just following the biology or just following the nanomaterial science. So that is the first part of that story, how surface engineered nanoparticles helps us to understand the biology of cancer better. And with that understanding, we identified MECO1 is a glycolytic switch in ovarian cancer. So next one is also another interesting story, because and again, this is receptor targeted nanoparticles. And the questions that we wanted to ask there is, so when we attach a targeting agent to a nanoparticle, so do the targeting agents behave similarly? Meaning, do they endocytose using the same pathway as the free targeting agent, or they change their pathways? And so that's what we wanted to know. And we designed nanoparticles having different number of targeting agents and wanted to understand how they're going to internalize into the cells. So in that same time, again, it started in early 2008, 2009, we were also working on another hypothesis. And that hypothesis was the macropinocytosis. So we were thinking the metastatic cancer cells in particular, they have a different or very high metabolic demand because they want to move from one place to the other. So they are not like the primary cells, they're sitting within the tumor and they do not want to move because they probably have plenty of food supply. But for the metastatic cells, then since they'll have to move from one place to the other, they probably need much more energy. And as you know, energy comes from the food. So we thought those metastatic cells probably are hungry cells, and they will have a different food uptake mechanism as compared to the primary cells. And so when we are hungry and when we are starving, so what do? And if we find food, we just basically gulp those food. You probably stretch both of your hands and eat them as quickly, as fast, and as much as possible. But obviously the cells, they do not have hands like we have, but they have their membrane. So what they do, they basically mobilize their membrane and gulp whatever surrounding them. And that's the process called cell drinking process, or it's also called the process of macropinocytosis. So we thought that malignant cells probably have some drinking problems. And so with that hypothesis, we just wanted to go back into the literature and then see if anybody has published anything along that line, whether macropinocytosis is linked to cancer. And to our surprise, we found this paper in 1937. So what they showed is that really the malignant cells, they do rely on macropinocytosis. So in this paper, what they were showing is, so they did like chemical induced carcinogenesis. They used 160 mouse tumor cell lines and then treated with this chemical carcinogen, and they found out that 90 out of 160, they basically utilizes macropinocytosis. So the process of macropinocytosis is prominent in those cells. So we now feel good. There is some literature evidence that macropinocytosis happens in cancer cells. So there is another paper that came out in 1980s, and that paper says those human synovial cells from rheumatoid arthritis patients, they use macropinocytosis. And basically you can differentiate between non-rheumatoid synovial cells versus rheumatoid diseased synovial cells from their macropinocytotic index. And non-synovial cells have lower macropinocytotic index than the rheumatoid arthritis, the synovial cells from the rheumatoid arthritis patients. So now we have some evidences that macropinocytosis is important not only for cancer but for other diseases as well. And if you remember, we are also trying to understand at the same time how really nanoparticles get into the cells. When we decorate the nanoparticle surface with different number of antibodies, what is the intracellular uptake pathway? So Sanjeev was working in my lab during the time 2009 as a postdoctoral fellow. Now he's a professor in a university in China. So he created these different nanoconjugates. This is gold nanoparticles of 5 nanometer in diameter, and he has put cetuximab, which is an anti-EGFR antibody, and in one case he put only one antibody, in other cases he had put like 3 antibodies. And surprisingly, what you have found, depending on the number of antibodies that you put on the nanoparticle surface, it can modulate the trafficking and the mechanism of antibody-induced receptor endocytosis. And again, we published this in 2010 in PNS. So one of the figures from the paper is that, so this is we call AUC225C. AUC225C means completely covered, and AUC225P means partially covered. And we have, again, tons of data to demonstrate that when you have 3 antibodies of cetuximab on a 5 nanometer gold surface, the non-specific uptake of those entire conjugate is completely blocked. Versus when you have partially conjugated nanomaterials, means when you have 1 antibody per 5 nanometer diameter of gold nanoparticles, then there is a lot of uptake which is not going through the EGFR mediated endocytosis process. So we proved that by overexpressing dynamin mutant. So, for those pinching of those membrane and 2 endocytos, those pinched membrane portion, you need to have these small GTPases, which is dynamin 2, which helps in pinching of the cellular membrane. This dynamin mutant K44A is a negative mutant, means dynamin function is completely inhibited. So if you use AUC225C, so now you have cells where you have overexpressed mutant dynamin, where GTPase function of dynamin is gone, and then you are treating with AUC225C, where the antibody gold is completely covered with the antibody. You see there is complete inhibition of that uptake pathway. So they're stuck at the membrane. But when you have AUC225P, where you have only 1 antibody per nanoparticle and the rest of the gold surface is free, you see this dynamin has nothing to do with the intracellular uptake. And we have proved that this AUC225C gets into the cells through dynamin 2 dependent cavular uptake pathway, whereas AUC225P gets into the cells through CDC42 dependent macropinocytosis pathway. And we have found these beautiful TM pictures where you can see how really this C225, which is just the antibody, and when you treat the cells with the antibody, this really nice grapefruit-like structure, which are cavular structure that you have captured in the TM microscope. It is really copybook type of images, luckily, that you have got. And as you can see in those cavular pit, these AUC225C, where again gold is completely covered with that antibody, is nicely taken up into the cavular pit and also in another cavular structure as well. So, but when you take AUC225P, which is partially covered, and this is what we have found, and this is how different types of macropinocytotic cup that is formed by the cells. As you can see in one of this cup, it just stretches the membrane out and close the loop, and exactly that kind of structure you have found through our nanoparticle treatment. It is exactly similar like it's found in the book. So we were so happy with this kind of TM pictures. Again, all of those things we have done at Mayo Clinic. And again, we published this paper in Angwantha Chemi in 2012. So now the important part of this finding is that now we have a nanoconjugate which gets into the cells through cavular uptake pathway, and you have another nanoconjugate which gets into the cells through macropinocytosis pathway. Then we thought, okay, now we know those information. Now why don't we utilize those information and then see whether we can find out some regulator of macropinocytosis in the cancer cells, so that if we can then disrupt or knock out that regulators, it might inhibit cancer cell growth. So what we did, we now basically used pull down assay. So basically we lysed the cells, say pancreatic cancer cells, we got the lysate proteins, incubated with AUC225C, completely covered gold with antibody, and AUC225P, partially covered gold with antibody, and then pull down proteins, and we did mass spec analysis. And then Sunao took up the job what Sanjeev left, and he's now a senior scientist in a company. And we did mass spec analysis. So what we have found is that there are 652 proteins which are common to both these conjugates, and 223 is exclusive for, this is partial, this is completely covered, and this is 338 is specific or exclusive for this partially covered, where you have one antibody per particle. So now the question is, out of these 338, any of them could be a target of macropinocytosis. So what we did then made some more stringent cutoff. Basically, how many peptide matching you are going to take for a positive confirmation. So we make that cutoff stringent to, if we select 10 peptide matching, then we will see, okay, that could be a reliable target, protein target that is identified by mass spec. So even when we did the stringent cutoff, we ended up with 65 proteins. So each of the 65 proteins could have a role in macropinocytosis, or they could not have a role in macropinocytosis. So how to select one or two proteins from that 65 proteins? So what we did, so again, during the time this article came up, too many roads not taken. Again, this article came up again 10 years after the completion of the Human Genome Project. And the crux of the article is, more than 75% of the protein research still focuses on the 10% of the proteins that are known before the genome was mapped, even though many more have been genetically linked to the disease. So basically, it tells us that there are a lot of proteins, still we do not know what is their functions are, because we have been working mostly on those, at least 75% of those proteins which are known even before the Human Genome Project. So interestingly, within that 65 proteins that we figured out from that high peptide cutoff, few of them we found out there was no report about them in the literature. So we thought, okay, let's then pick one of those proteins and then see whether it plays any role in macropinocytosis. And if we could demonstrate that unknown protein has something to do with macropinocytosis, then we are bringing a new molecule into the system, and it may also help us to find out a new regulator of macropinocytosis, and that may be used to inhibit tumor growth. So one such protein was Eubaptic, and it is ubiquitin binding associated protein 2. So what we did figure out is that the full-length version of the protein is like this. It has a UBA domain, and rest of the structure has no sequence similarity with any of the known proteins available so far. And during that time, there was no report about the protein in the literature. So we thought, okay, let's work on the protein and then see what we end up with. So as we always do, first we look at the expression of the proteins in different pancreatic cancer cell lines along with the normal cells. And we see Eubaptic expression is much lower in the normal cells, but it is much higher in the cancer cells. Also, interestingly, if you look at BXPC3 cells, and you see the expression is similar to kind of the normal cells. And the difference between BXPC and rest of the other pancreatic cancer cells are that BXPC3 has a RAS which is wild type, and rest of the cells have RAS which has RAS mutated. And as you know, in pancreatic cancer, in most of the cases, RAS is mutated. So we thought, okay, let's see if we knock down Eubaptic whether it really inhibits macropinocytosis or not. So dextran uptake is a readout of macropinocytosis. So in SI control, we treated the cells with dextran red, and with SI Eubaptic, we again treated with dextran red. And as you can see, there is dramatic inhibition of dextran red uptake by inhibition of macropinocytosis by Eubaptic. So it tells us Eubaptic is really a regulator of macropinocytosis. And when we used colopromazine, when we use transferrin, which goes through clathrin-mediated uptake pathway, and as you can see, Eubaptic knockout has nothing to do with transferrin uptake, but when you use chloropromazine, which inhibits clathrin-mediated uptake, we see robust inhibition of transferrin uptake into the cells. So it tells us that Eubaptic is really specific for macropinocytosis, and it does not alter any other uptake pathways. So we started there, and then again we did it in other cell lines as well. We did find the similar effect. It is Eubaptic is inhibiting macropinocytosis in other cell types as well. And then we also looked at the expression of Eubaptic in tumor tissues, and we found that in PDAC pancreatic ductal adenocarcinoma, it is again highly overexpressed in tumor tissues, and that correlates with poor prognosis. And then we went on to prove that when you knock down Eubaptic in vivo, there is robust inhibition of pancreatic tumor growth. And also you showed by knocking down Eubaptic, you can also inhibit macropinocytosis in vivo as well. And then but the next question is, okay, what is the mechanism? Eubaptic is inhibiting macropinocytosis, but how? And this paper came out in 2013, which says RAS transformed cells, they utilize macropinocytosis for their survival. So we started the work in 2019, and this paper came out in 2013, but we still feel good. Okay, maybe we have found out something which regulates KRAS. And that's what actually I found. When we knock down Eubaptic, you see KRAS activation goes down in BXPC3 cells as well as SPC1 and PANC1 cells. And then, of course, we deciphered all the molecular mechanisms through a lot of rescue experiments in vivo studies. And we figured out Eubaptic regulates macropinocytosis actually by regulating KRAS activation. So this part of the story, as you can see, we tried to understand how nanoparticles having different targeting molecules will enter into the cell, will lead us to identification of a novel molecule, which is Eubaptic, which regulates macropinocytosis and KRAS activation in pancreatic cancer. I think that is the beauty of nano. When you try to understand how really nanomaterials enter into the cells, interact with the cells, it really gives us something which is very, very unique, which otherwise we would not have been able to discover. Like Eubaptic would know, like Eubaptic could be a molecule which would be a regulator of macropinocytosis. So that's the second part of the story. And the third part, and again, we didn't stop here. Now we have generated Eubaptic knockout mouse models, and we are now trying to figure out whether Eubaptic is playing a role in pancreatic tumorigenesis, how pancreatic tumor evolves. And we have KPC mouse model that people use. So we are basically using our Eubaptic mouse model, breeding with KPC mouse model, and trying to understand whether Eubaptic is involved in pancreatic cancer tumorigenesis. So next part is, I'll go quickly, it's our self-therapeutic nanoparticles. So normally, what we think nanoparticles is as a vehicle. But in 2004 and 2005, we figured out, or we actually serendipitously found out, that gold nanoparticles in particular, they have anti-angiogenic property. And so what also so these cancer cells, cancer-associated fibroblasts, and endothelial cells, they secrete like lot of angiogenic factors, and that's how they crosstalk with each other. As for example, the TGF beta, SDF, IGF, and BGF, and all of those the cancer cells secrete, and they are angiogenic in nature, and they have, all of them have heparin binding domain, and those molecules are involved in crosstalking with each other. Like cancer cells crosstalk with cancer-associated fibroblasts, they crosstalk with endothelial cells, and there is a perpetual crosstalk that goes on within the tumor microenvironment, because those cells are secreting a lot of factors. And so you'll be like surprised to know cancer cells actually secrete around 1100 molecules into the conditioned media, and similarly the cancer-associated fibroblasts. But the challenge there is, okay, which one is the key perpetrator, and whether nano can be used to identify the key perpetrator and then disrupt this multicellular crosstalk that goes on in the tumor microenvironment. So as I said, we found in 2004, 2005, gold has anti-angiogenic property, and the way it performs it inhibits the function of the heparin binding growth factor by binding through the heparin binding domain. And what are those heparin binding growth factors? Most commonly studied are VEGF165, and then FGF2, and then PLGF, PDGF. There are a lot of those growth factors which have like heparin binding domain. So when gold bind to those proteins through that heparin binding domain, it inhibits their function. And what we have found is that if you take gold nanoparticle of 5 nanometer, 10 nanometer, and 20 nanometer size, the 20 nanometer actually has the highest stability to inhibit the function of the heparin binding growth factors. Whereas, and again, this is an example where, again, FGF2 or BFGF, basic fibroblast growth factor, and you are incubating with gold and treating fibroblast cells with that gold and the growth factor conjugate, and you see a robust inhibition of proliferation of the fibroblast cells. Whereas EGF, which do not have, or which does not have an heparin binding domain, it has no effect whether you put them on gold surfaces or not. Basically, gold does not interfere with the function of non-heparin binding proteins, but it only interferes with the function of the heparin binding proteins. And the way it does, and this is again, we did it with Vince Rotello, we did like circular dichroism study. So we thought maybe interaction of the protein with gold through that heparin binding domain is changing their conformation. And that's what we studied here. If you look at the native FGF structure, the structure is heavily perturbed when you incubate that with gold nanoparticles and ran the CD spectrum. Whereas with the EGF, you see there is virtually no change in the protein conformation or the secondary structure of EGF when you incubate that with gold. So gold basically binds through the heparin binding domain of those proteins and then inhibit their function. And as I've told you, since there are a lot of proteins, the lot of proteins which are secreted by those cancer cells and cancer-associated fibroblasts have heparin binding domains. So we thought whether we can modulate tumor microenvironment by using gold nanoparticles. And again, we published this paper in PNAS in 2013, and there we coined actually the term self-therapeutic nanoparticles by just exploiting the unique ability of gold nanoparticles to bind to heparin binding domain containing protein and inhibiting their function. And also in pancreatic cancer, we showed it can really disrupt the crosstalk between cancer cells and cancer-associated fibroblasts and inhibit tumor growth. And as you can see in this accompanying picture that you have published in this essays, Applied Materials, I think 2019 or 2020, when you treat these fibroblast cells with the gold nanoparticles, there is lipid droplet accumulation. And lipid droplet accumulation is the indication of the fibroblasts is transforming themselves into quiescence. And that we have figured out the mechanisms in this paper. So again, we use this concept. Now this is fresh from the pot. It came out last week, that disabling partners in crime. We basically used gold nanoparticles, and we showed that in this particular paper that it can disrupt the crosstalk between cancer cells, cancer-associated fibroblasts, and endothelial cells. And we figured out what is the underlying molecular mechanism and what is the key perpetrator, which is actually doing the crosstalk among those multicellular environment. So with that, but again, probably I'll have to skip. So now the question is, okay, what are the proteins that gold are binding to? So we do like a lot of protein corona studies. So we wanted to understand what are the proteins that gold are capturing, and those are the proteins which may be responsible for the tumor growth and metastasis. And we published the detailed protein corona evolution in 2014, this bioconjugate chemistry paper. And yeah, so basically, let's go here. So what we did, we again got the cell lysates and then incubated with nanoparticles with different time, like 5 minutes, 15 minutes, 1 hour, 2 hours, 6 hours, and 24 hours in normal cells and cancer cells. And we compare what are the proteins that are captured in the cell types. So if you compare this Venn diagram, as you can see, if you compare the lysate proteins from those pure cancer cell normal cell lysates, which is OHN 82780, versus when they're captured on the nanoparticle surface. So you see at 6-hour time point, these 41 proteins are very exclusive for the cancer cells, and it is not found out anywhere else, not only in the cancer cell lysate, not only in the normal cell lysates, but it is found only when the nanoparticles are used. So basically, these are the proteins which might be low abundance proteins, which could not be detected through normal mass spec facilities or mass spec approaches. Similarly, at 24-hour time point, we see like 65 proteins which are exclusive for the gold captured cancer cell lysates only, which is not present in the normal cell lysates, either from the normal cells or the cancer cells. Now, these 65 proteins actually could be therapeutic target as well, because they're very specific for cancer. But how do you go after them? So we do this kind of functional nodal networking based on co-expression, co-localization, shared protein domain, and we create this kind of functional nodes. This bigger node means this protein is highly connected and connected with other proteins in the network, and whereas this smaller node means they're singularly connected or least connected in the network. So our thought process was, if you disrupt a node like this, you might get a better effect, because it might disrupt the function of the other proteins that are connected to it. So that's how it started. And actually, we actually selected a couple of proteins from here which are highly connected versus some which are not so highly connected. And what we are showing here is that these two proteins which are highly connected, if you knock them down, you can see like robust reduction of cancer cell proliferation versus this protein which are not so connected to the network. So this gives again an idea, those cell therapeutic nanoparticles and how it really interacts with the proteins and the cells. It gives an idea about what could be the molecular targets which are executing the perpetual crosstalk in the tumor microenvironment. So with these three stories, I guess I have been able to tell, or I have been able to convince you that if you really try to understand how nanoparticles interact with the cells, you end up with something which is very, very unique, which otherwise we would not have been able to discover. And we also do, I'm not going to go into it, like next generation of base RNA delivery system, we created these hybrid nanoparticles by doping conventional liposome with gold, and we have shown it's much more efficient in gene silencing efficacy than what is commercially available now. Again, we published this paper in Science Advances a couple of years ago. And basically, doping of this gold into the liposomal formulation actually switches is intracellular uptake pathway, and that's how it enhances its therapeutic efficacy. And with that, I think, so these are my group. There are some additions to the group as well. And these are my current active grant funding. I think I'm lucky so far. I've been maintaining these multiple R1 grants over the last 12 to 13 years. And yeah, and again, thank you all for your kind attention.
Thank you for this great talk, Professor Mukherjee. Several questions came in. I'll read them. Based on the calcium entry, do you think you may have effects on ER or UPR, which is involved in proliferation and apoptosis?
Right. Yes, you were right, and we have seen that there is effect on UPR, and that's the area, or that is we have published in the JBC paper in 2013. Yes, we have looked at that like UPR effect, unfolded protein response effect, and how really MIC1 affects the UPR. Yeah, we have published that in the JBC paper.
Thank you. Great talk. Is there any role of calcium influx with endoplasmic reticulum stress, which is related to microRNA expression come out via in exosomes related to cancer?
Right. I think ER plays a very big role, and actually, so I have not had the opportunity to talk of different areas that we work on. So we are also interested in looking at organelle crosstalk, right? How so right now I was talking about multicellular crosstalk, how cells are talking to each other. We are also interested how organelles within the cells are talking to each other. Now, ER and mitochondria is actually, again, I will say partners in crime, because those calcium tunneling from ER to the mitochondria, which is a very, very critical phenomena to make cells either survive or to make cells undergo death. So yes, there is a huge interest in that my ER mitochondria crosstalk through the calcium tunneling, and we are actually right now identifying, it's called MAM, mitochondria associated membrane. So which part of the ER is actually tethered to which part of the mitochondria, because through which that calcium tunneling is taking place, and what are the molecular mechanisms that are tethering ER and mitochondria. So that's a huge and upcoming area of research. So we are working into that. Yes, that ER calcium plays a big role in the cellular physiology, particularly in glycolysis as well as in oxidative phosphorylation by regulation of that calcium tunneling.
Thank you. And the other person is asking, which pathway should be better for better therapy? Peptide or antibody therapy?
Right. Again, I will say, so there is not a single pathway. It has to be a combination of many different things. So again, as you know, like antibody is a viable therapy. So there are a lot of antibodies that nowadays are in the clinic, including bevacizumab, cetuximab. There are a lot of other mAbs as well. But again, for the peptide, again, antibody has its own problems. So are the peptides. For the peptide, the problem is how really you transport it to the cells. You need to modify the peptides, and you need to prevent the degradation of the peptides into the circulation and all of that. But each therapeutic system, they have their own advantages as well as their own challenges. But I don't think it is yes or no, but we have to see which system actually works for which diseases, and what type of system that we are using at. So it is, I cannot say whether this peptide is better versus antibody is better. It really depends on the context that we are talking about and the diseases that we are talking about. But each of them are viable. The antibody is viable, as you know, it has been there in the clinic for a long time. And now this peptide and other small molecules are also coming up.
Thank you. Final question. Can we do early diagnosis of cancer by ER pathway?
It is possible. It is possible, but again, as we know, early diagnosis has always been a problem, in the sense that there is not many good markers which were really detected at very early stage of the disease. But I will say this protein corona approaches has, I will say, better possibilities of identifying early biomarker for any diseases, not only in cancer. And as you can see from the protein corona studies that we have been doing and other people have also been doing, is that you can make invisible things visible. So one thing we all know, Mother Nature is very, very clever. So what they do, they dump in a lot of unnecessary materials to basically protect or hide those important molecules. As for example, if you look at the serum, there are 12 different, high abundant proteins which basically hides or mask a lot of those low abundance proteins. But using this protein corona approach and following up with the patient samples, I think there is a better chance of identifying those early detection biomarker, not only in cancer, but also in other diseases.
Thank you. Thank you so much for this fantastic talk. That's all the questions I have, Professor Mukherjee.
Thank you. Thank you, Mehmet, and thank you, everyone. And again, if you guys have anything in mind, just reach out to me through email, through phone call, and I'll be happy to communicate with you, collaborate with you if any opportunity arises. And as I was talking to Mehmet, that I like in-person meeting more than the online because the interaction, the bonding is much better with in-person than through Zoom.
Thank you. Have a great day, sir.
All right. Thank you so much.